The shape of an endoplasmic reticulum subcompartment is determined by a number of proteins on the exterior, which can determine whether the ER has a sheet-like shape or a rounder, more tubular shape. In certain areas of smooth ER, known as ER exit sites or ERES, there are proteins which induce a curvature in the region and allow vesicles to form. Separate proteins form the inner coat and outer coat as vesicles bud. As other proteins interact with the proteins forming the vesicles, they can transport the proteins they have attached into the vesicle. There are many proteins which will transport individual molecules. For example, the protein Tango 1 transports pro-collagen into a vesicle so that it can ultimately be transported and form collagen outside the cell. In the ER to Golgi intermediate compartment, or Ergic, properly folded proteins are packaged into vesicles so that they can be transported to the Golgi for further maturation and folding and ultimately secretion from the cell or transport within the cell. Not only will vesicles form which travel to the Golgi, vesicles from the Golgi will also return to this region of the ER. If either of these forms of transport are inhibited, the anterograde transport traveling to the Golgi or the retrograde transport traveling from the Golgi, then products will accumulate and the Golgi body will ultimately be disassembled. The region of SER, known as Erqc, or ER Quality Control Compartment, is tasked with identifying the misfolded proteins and either attempting to refold them and send them on to the Golgi for further processing, or to target them for destruction if they are terminally misfolded. If they are targeted for destruction, they will bind a protein known as ubiquitin and undergo retrotranslocation to pass through a pore out of the Erqc out into the cytoplasm, where they will be destroyed at a proteasome which digests protein. This destruction of misfolded proteins is known as ERAD, or ER-associated degradation.